Hello, and welcome to your fourth and final lecture for Chapter 5, Biological Molecules. Up to this point, we have discussed the first three categories of biological molecules: carbohydrates, lipids, and proteins. Today, we will briefly introduce the fourth class of biological molecules called nucleic acids, which are important for storing and transmitting hereditary information. You can think of the hereditary information as a blueprint or a recipe for everything that your cells and your body can do, for the way you look, for who you are. The information has to be somewhere, and it's held in your DNA, one type of nucleic acid. As we discussed previously, proteins make up the bulk of an organism. The major biological molecule of cells is that of the protein. We talked about polypeptides being a polymer that is then folded into a protein. So we are mostly protein, but where does the information come from to build these proteins? Well, the amino acid sequence of every polypeptide, and thus every protein, is programmed by genes. And genes are simply stretches of DNA, one of the nucleic acids you need to know. If this is a DNA molecule, which is always double-stranded and held together by hydrogen bonds, you could have a gene here and another gene here. And as you went down that DNA molecule, you might find 1,000 or more genes. So a gene is simply a stretch of DNA that has information, and often it's information for a polypeptide of some sort. There are two types of nucleic acids: deoxyribonucleic acid, or DNA for short, and ribonucleic acid, which is related, RNA. The names of these two nucleic acids are derived from the sugars that we find in their monomers. I'll show you these in a little bit, but for now, DNA monomers, which are called nucleotides, have the sugar deoxyribose, whereas RNA monomers, also called nucleotides, have the sugar ribose. Notice the "-ose on each name? That's consistent for sugars. So deoxyribose leads to deoxyribonucleic acid, whereas in RNA, the ribose sugar leads to ribonucleic acid. So the sugars found in their nucleotide monomers are used in their names. DNA, the largest of the nucleic acids, provides directions for its own replication. And when do you think DNA needs to be replicated? Well, regardless of the organism you're talking about, this is an important step in cell division. If a cell is going to split into two daughter cells, it must first copy all of its DNA so that both daughter cells get the DNA complement, so that both daughter cells get the full genetic complement, all of the information necessary. Not only does DNA provide directions for copying itself, but DNA directs the synthesis of other molecules in the cell. For example, RNA. DNA directs the synthesis for all types of RNA, and there are several different types of RNA based on what it does for the cell. One type of RNA is messenger RNA, or mRNA for short. And mRNA is important for directing protein synthesis, which we mentioned at the very beginning of this lecture. Protein synthesis occurs in the ribosomes. So we have a stepwise flow of information when we're making our proteins. It starts with a gene at the level of DNA, which is used to build a molecule called mRNA, which is then used to build a protein. This stepwise flow of information to build a protein is illustrated in the following slides. What type of cell are we looking at in this picture? Is it a eukaryote or a prokaryote? If you notice there's a nucleus, you should have said eukaryote. In a eukaryote, DNA is always housed in the nucleus and it never leaves. Therefore, DNA replication always happens in the nucleus, and the first step in gene expression, which we're about to talk about, always happens in the nucleus. DNA is always going to be found there in a eukaryote. In the first step of gene expression, the flow of information moves from the level of a gene to the level of messenger RNA. So as you look at DNA, you'll see that it's a double-stranded molecule, and recall that the two strands are held together by hydrogen bonding. In this illustration, if we're looking at one gene here, only one of the two strands of DNA is important for making a protein. And that strand is called the template strand. So let's just say that this DNA strand right here is the template. On that template, you find a string of letters, or bases, A's, T's, G's, and C's. And that string of bases is used to direct the synthesis of messenger RNA so that a complementary messenger RNA is constructed from the gene. We will discuss complementarity in a little bit, but for now consider this. If the DNA template strand has an A, then the complementary messenger RNA will have a U. If the DNA template has a G, the messenger RNA will have a C. If the DNA template has a C, the messenger RNA will have a G. And if the DNA template has a T, the messenger RNA will have an A. So the base sequence of the newly formed messenger RNA is not identical to the DNA, and it's not identical to the gene, but it's complementary. This is the first step in gene expression, is building a complementary messenger RNA from the template strand of a gene in the DNA. And of course this happens in the nucleus because that's where the DNA is. That's where the blueprint is, or the recipe. After the mRNA is made, it has to leave the nucleus through a nuclear pore. These are transmembrane proteins. They span the entire double membrane of the nuclear envelope. The RNA is able to leave through the nuclear pore and enter the cytoplasm. And it's important for the mRNA to go to the cytoplasm because that's where the ribosomes are found. And ribosomes do the next step in gene expression. In the cytoplasm, ribosomes will dock on the messenger RNA molecule that was just made. And you can see the ribosome has two subunits. It has a large subunit and a small subunit that basically make a little sandwich out of the messenger RNA molecule. The ribosome then moves along the messenger RNA. It pulls it through like a tape. And as the ribosome is moving along the messenger RNA molecule, it is reading the base sequence of that mRNA. In other words, it's reading the A's, the U's, the G's, the C's. And it's using that information to decide which amino acids are joined together to build the polypeptide. In other words, the ribosome is translating the genetic code. So here you can see the genetic code is found on the mRNA molecule, and it consists of a string of letters or bases. So this basic flow of information is how the gene is used to make the protein. This is called gene expression. We will now talk about the structure of nucleic acids. You have to know a bit about the structure in order to understand how they function, because as always, structure determines function. Nucleic acids are polymers. They are very large molecules or macromolecules, and therefore they are called polysomething. In this case, we refer to nucleic acids as polynucleotides because they are made of many nucleotides joined together. So based on the name of the polymer, you should be able to guess what the name of the monomer is. The monomers for all nucleic acids are nucleotides. Therefore, when we join several thousand or even several million nucleotides together, we build a polymer called a polynucleotide. Let's move to the next slide, and then we will return to this one to visit that concept of the nucleotide again. Going back to that monomer versus polymer relationship. As mentioned on the previous slide, if we're talking about nucleic acids, the monomers are nucleotides. If we have several thousand, or in the case of DNA, several million dehydration synthesis reactions, we can build the polymer, which is called a polynucleotide. Or we can say it is a nucleic acid. Either term is fine for the polymer. It's a polynucleotide because it's made of many nucleotides joined by covalent bonds, or we can say it's a nucleic acid. Those terms are interchangeable for the polymer in this case. There are two types of polynucleotides or nucleic acids, and so those specific examples are DNA, or the other example is RNA. These are very similar polynucleotides. However, there are some differences. Let's begin by similarities. They're both built from monomers called nucleotides, and the basic structure for a nucleotide is a phosphate group. I'm just going to abbreviate that with a P for now, but you should know that a phosphate group has a phosphorus atom and 4 oxygens with negative charges. That is attached to a 5-carbon sugar. So there's an oxygen here in the 5-carbon sugar, and then there's 1 carbon, 2 carbon, 3 carbon, 4 carbon, and 5 carbon for that sugar. Because this sugar has 5 carbons, we can say it is a pentose. And then that 5-carbon sugar is attached to a base of some sort. That is the basic structure for the nucleotide monomers. A phosphate, PO4, 2 minus, a pentose sugar, and a base. So both DNA and RNA are made out of nucleotides, but the nucleotides can vary a little bit. In the case of DNA, the sugar is deoxyribose. However, in the case of RNA, this pentose sugar is ribose. They're almost the same. There is just a single oxygen difference, which I will show you in a moment. Another difference can be seen in the bases. DNA nucleotides can have bases that are either A, T, G, or C. So every DNA nucleotide has one base on it that is either A, T, G, or C. However, RNA nucleotides have one base on each of them that is either A, U, G, or C. So the difference in the bases is found in the thymine-uracil conversion. Thymine is only found in DNA nucleotides, whereas uracil is only found in RNA nucleotides. But A, G, and C, adenine, guanine, and cytosine, are found in both types of nucleotides. Let's go back to the previous slide. Continuing our discussion of the monomers. In the case of nucleic acids, our monomers are called nucleotides. And I already went through this basic structure with you. Every nucleotide has a nitrogenous base. This just means it contains nitrogen. Recall that that base can be A, adenine, T, thymine, G, guanine, or C, cytosine, if we're talking about DNA nucleotides. And that base is going to be A, adenine, U, uracil, or G, guanine, or C, cytosine, if we're talking about RNA nucleotides. Every nucleotide also has a pentose sugar. So recall that a pentose is a 5-carbon sugar. And that sugar is going to be deoxyribose, if we're talking about DNA. And that sugar is going to be ribose, if we're talking about RNA. And then finally, every nucleotide monomer has a phosphate group, which consists of a phosphorus atom and 4 oxygen atoms, as well as a couple negative charges. So the basic structure of a nucleotide has a phosphate group attached to a pentose sugar, ribose or deoxyribose, and a base. That's your basic structure of your nucleotide. On the left side of this figure, we are looking at a small portion of a polynucleotide. Several nucleotides joined together by covalent bonds. Here is a nucleotide, here is a nucleotide, and another, and a fourth. So 4 nucleotides are joined together by strong covalent bonds that make up the sugar phosphate backbone. So sugar, phosphate, sugar, phosphate, sugar, phosphate, etc. There are strong covalent bonds that make up this backbone of our polynucleotides, whether DNA or RNA is being considered. If we zoom in on one of these nucleotides, we can see the 3 components that I went over previously: the phosphate group with its negative charge, the sugar, which has 5 carbons, 1, 2, 3, 4, and 5. And then finally, the nitrogenous base. Nitrogenous because it contains nitrogen atoms. The base options are shown to the right. So we've got different options for bases here. Bases can be called pyrimidines, or they can be called purines, and it depends on their ring structure. Pyrimidines have a single ring structure, and these include cytosine, thymine, and uracil, C, T, and U. Purines have a double ring structure, and they are adenine and guanine. So every nucleotide has one of these bases attached to it. The pentose sugar can be deoxyribose if we're talking about DNA, or ribose if we're talking about RNA. And these pentose sugars are identical except for this oxygen on the second carbon atom. Ribose has a hydroxyl group here attached to this second carbon atom, whereas deoxyribose has a single hydrogen atom. So the only difference between these sugars is that the one on the left is missing an oxygen, and the name suggests that. It's not ribose, it's deoxyribose, minus an oxygen ribose, unlike ribose, which has that oxygen. Taking a closer look at those pentose sugars, deoxyribose is shown on the left deoxy, so minus an oxygen, ribose. There is no oxygen there. Versus ribose, which has the oxygen on that second carbon atom. Besides that, the sugars are the same. And as mentioned, the bases come in two forms, two basic forms. Pyrimidines, which have a single six-membered ring. Those include cytosine, thymine, and uracil. Recall that thymine is only found in DNA, and uracil is only found in RNA. And the purine bases have two rings, a six-membered ring and a five-membered ring. And these larger bases include adenine and guanine. Cytosine, adenine, and guanine are found in both DNA and RNA. Now that you know a bit about the nucleotide monomers, you can better understand how they join together to form a polymer, which is called a polynucleotide, built from many nucleotides. In other words, a nucleic acid. Nucleic acids are built from thousands or even millions of nucleotides joined together. The process of joining nucleotides together is the same as you learned for the other three classes of biological molecules. Dehydration synthesis is the type of reaction that builds these polymers. So adjacent nucleotides are joined by new covalent bonds when the hydroxyl group on the third carbon of one nucleotide sugar reacts with the phosphate group on the fifth carbon of the next nucleotide. In effect, we are going to lose one molecule of water for every new covalent bond that forms between nucleotides, just like you've seen with every dehydration synthesis reaction. And this creates a backbone of nucleic acids that consists of sugar phosphate units.